Well, this title you can read for yourselves. It's it may be slightly grandiose considering the data I'm going to show you. But okay. So lichens, that's something that we've been lichens are organisms that we've been studying quite a bit in my lab or in our labs here in Akureyri for a while now, and they are very interesting models in many ways. They are symbiotic associations, as most of you probably know. By their very nature, they are strongly dependent upon each other. The fungal component of the lichen and the algal component, the so-called microbiont and photobiont. The photobionts being either green algae or cyanobacteria, what people used to call blue-green algae. Okay, and yeah, as I said, they're completely reliant upon each other, but also they rely upon their associated microbiomes, just yeah, like we all do, really. I mean, we all have all sorts of bacteria, like for example in our guts, that we rely upon for various things, and it's the same, of course, for lichens, and perhaps even more so. There is a lot of bacteria in lichens. We're talking about roughly about a billion bacterial cells per gram of lichen thallus. Okay, you may have seen in the title the name dog lichen. Dog lichen is just the common English word for lichens of the genus Peltigera or Peltigera, however you want to pronounce that. They are very common here in Iceland and elsewhere in the Arctic, subarctic, and also down into the more temperate zones. They yeah, as I said, they're very common indeed, even though people often don't notice them, which is a shame because they are very important. But they're not maybe they don't stick out like trees or flowers. But yeah, they are very important, and they are when you see a lot of lichens in a habitat, it makes you happy because you know that that's an indicator of good habitat health. They are generally fairly sensitive to pollution, for example. Yeah, so they are indicators of a healthy environment. Yeah, for the Peltigeras, there are 24 species of them here in Iceland. And as I said, they're very common. I just stepped out my house for half an hour to take these pictures of different lichens for you, or different Peltigera lichens. Okay, now I'm a microbiologist. So as Sean just told us, we believe that microbes, bacteria in particular, are the most important organisms on the planet. And so that's what I study in the lichens, is the bacteria and their microbiome. So the community of bacteria present in the lichens. I realize that most of you here in the audience are probably not very familiar with all of these Latin names that we use. So I'm going to try to not talk too much in those terms. But I'll just tell you that in Peltigera we see, as I said, we've been studying them for a while now, and we see fairly consistently the same species, the same types of bacteria being dominant within the lichens. And these are basically the alpha proteobacteria and a few others. Here is a nice little picture from a paper by Tomislav Chernava, where he sort of considers what the roles of these different bacteria are within the lichen, what they're doing there, how they are useful to the lichen, and so on. I'm not going to, we don't have the time, I think, to go into that in any sort of detail, but it is something that we're very interested in and have been studying for a while. If I have the time, I may come back to this towards the end. Now, but so what on earth has all this got to do with climate change and and sustainability? Well, so just in general, bacteria are we can consider bacteria as good indicators of environmental change. They are they unlike us and unlike other multicellular organisms. They are completely exposed to the elements wherever they are. They don't have a skin, they don't have a bark or a cuticle or anything of that sort. They're single-celled organisms. So how they deal with stress is basically uh, becoming dormant and hoping that the conditions will become favorable again. Um, and so it stands to reason that the composition of the microbiome in whatever system you're looking at will be subject to change according to environmental conditions such as climate. And indeed, that turns out to be the case. Here is a paper from our myself and my collaborators from a couple of years ago, where we were looking at at soil microbiomes and how they change in response to, as it says there, extreme events. We're talking about like heat waves, like floods, like droughts, all these climatic events that we are told are going to become more of a problem as the climate warms. And indeed, we found that there were, as it says there in the title, predictable responses to these events in terms of the composition of the bacterial and the fungal microbiomes, but also in terms of their activity, as we can see here. Well, I know you can't really read this picture, but just trust me when I tell you that it shows that there are differences in the activity as judged by the proteome. So the proteins present at any given time in all of in the whole collection of the microbiome. Um, so bacteria are in fact good indicators of environmental change. Now, let's go back to the lichens then. A couple of years ago, well, a few years ago, time flies. Ingeborg there did her PhD in my lab, and one of the things that she did was to compare the microbiomes in this lichen, Cetraria Icelandica, and also in this moss here, Raccometrium anuginosum, compared their microbiomes and some of the activities within these organisms, specifically nitrogen fixation. Compared that within or inside and outside these chambers here, these chambers that simulate warming and have been in place up in Akureyri now for I think about 25 years. Um, and and she found that yes, indeed, I'm not going to show you the data because we don't have the time, but yes, indeed, there are some very clear differences and some less clear differences between the microbiomes of these mosses and lichens inside and outside the boxes. You can just read her papers if you want the data. But what the take home message is that it is a lot more complicated than there just being a straightforward response to the increased temperature. In fact, what you also see happening in the boxes, of course, I mean, there will be an increased growth. Well, now you can't see the box anymore, but there is increased growth for example, of betula and other plants. So you have more plant detritus. Yes. Oh, yeah, time. Yeah, sure. Thank you. So there's more detritus and so on. So the environment has changed. It's we would still consider this the same habitat type according to the definitions of Náttúrufræðistofnun, but there have been market changes. So the microbiomes are responding to more than just the temperature. But we're stubborn, and we are convinced that we can read something important from the lichen microbiomes if we compare them across a larger spectrum, so to speak. So we got a small grant for like a preliminary study. I do not want to warn you about that. It's what you're going to see in the coming slides is very preliminary. Uh, where we compared the dog lichens, coming back to them again, in Icelandic habitats, we go all the way from the very Arctic tundra environment in Langanes and to a temperate rainforest here in Cumbria, and various different habitat types in between. Sampled a bunch of peltigeras of various species and compared the composition of the microbiomes as well as their metabolomes, which is another way of assessing their activity, as I'll get to in just a second. Yeah, we have a bunch of different peltigera species. Um, One thing I want to point out real quick, even though time is flying, is that when you get into the field and start collecting samples, you always see something interesting, so you get sort of sidetracked into different things. And we saw, particularly in Scotland, a lot of diseased lichens. They looked unhealthy. So we sampled some of those as well. You'll see why I say this in just a moment. But anyway, what I wanted to point out here is just, okay, we have a bunch of different peltigera species from a bunch of different environments, so we can do all sorts of comparisons. Whoops, what's not responding? There we go. And now the metabolome is basically just the entire set of metabolites present at any given time, and we assess that. I absolutely do not have the time to go into detail about the methods we used, but basically, so there are 16,191 different metabolites that we are looking at in the samples. And when we try to assess whether there are differences in the compositions of these 16,000 metabolites, yes, indeed, of course, there are. Most clearly, most obviously, with the different peltigera species. We probably could have expected that beforehand. But what really surprised me was that those diseased lichens that we sampled were basically indistinguishable from the healthy ones. That was quite surprising. Uh, Now, but we also can see some differences in the different habitats. So we try to group those habitats that we sampled, and there were, I think, about 12 of them, if I remember correctly. Um, of course, as you can see when you look at the picture, it's not really easy to read trend from this, and that wasn't really the point anyway. But yeah, we do find a difference between the different habitats, and that turns out to be even more pronounced than the difference between the regions, between Iceland and the UK. Perhaps sort of reminiscent of what Ingeborg saw in her study. However, uh, we do see a surprisingly pronounced difference in some when we look at particular compounds. For example, the Icelandic Peltigera membranacea hardly produces any usnic acid at all, whereas the UK ones do. The usnic acid is a secondary metabolite of the fungus, which it produces to protect itself against bacterial pathogens that can make the lichen sick. So, yeah, maybe just because the UK lichens are sick, that's why. Okay, now I see I've run out of time already. I'm sorry about that, but yeah, we did also compare the composition of the microbiomes more directly using just the DNA sequencing, and basically, yeah, it's basically a similar thing. When we look at the microbiome, we also see similar differences, I suppose, as we do from the metabolome. I'm not going to go into this at all. Just because I'm a microbiologist, I like growing bacteria. So just for fun, well, not just for fun, because we plan to use these bacteria in future research, but we've isolated a bunch of the bacteria that we can tell from the microbiome should be present. So yeah, we can grow them and hopefully poke them with a stick and find out something interesting. So yeah, that's it. Thank you very much.